Gram-positive bacteria are often the ones students feel most comfortable with. Lots of familiar names, lots of classic diseases. But I want us to approach this differently. Rather than memorizing a list of names and associated diseases, we're going to group these bacteria by what they do, where they live on the host, how they cause disease, and what that means for our patients. By the end, you should be able to walk into a clinical case and think logically about which gram-positive organisms belong on your differential list. Gram stain morphology is not just taxonomy. It narrows your differential list immediately. If you see gram-positive cocci in clusters on a wound swab smear, you're thinking Staphylococcus before culture results are back. Chains in a joint fluid? Streptococcus. Large gram-positive frauds in necrotic tissue? The spore formers jump to mind. Clostridia. The morphology is data. And combining it with clinical context, the species, the site, the nature of the lesion, gets you to a working diagnosis fast. Cocci are consummate opportunists. They live peacefully on your patient's skin and mucosae all the time. They only cause disease when something goes wrong: a wound, a surgical incision, a catheter, a weakened immune system. And when they do invade, they come armed. Coagulase is particularly clever. It converts fibrinogen to fibrin around the bacterial colony, forming a physical shield that neutrophils can't easily penetrate. Protein A binds the Fc region of IgG, the wrong end, essentially disabling opsonization. These aren't random features, they're evolved countermeasures to the very defenses we rely on. And MRSPN drugs, methicillin-resistant S. pseudo-intermedius, same resistance mechanism as MRSA, different species, big clinical problem in companion animals. The hemolysis pattern on blood agar is a fast, practical classification tool. Beta hemolytic streptococci, the complete hemolytics, are generally your most virulent. Streptococcus equi is a classic example. It causes strangles in horses, a highly contagious disease characterized by abscess formation in the lymph nodes of the head and neck. It's so well adapted to horses that it has essentially lost the ability to survive outside the host, but it spreads efficiently through direct contact and fomites within a herd. S. suis in pigs is worth flagging as a zoonosis. Farm workers and veterinarians can develop meningitis from exposure. That's a serious clinical and occupational health concern. Clostridia are a remarkable group because they are fundamentally toxin factories. The organism itself often does relatively little. It's what it secretes that kills. And the ecological key to their success is a spore. Clostridium Chauvii, the cause of blackleg in cattle, lives as spores in the soil for decades. A calf grazes and ingests spores. The spores germinate in anaerobic tissue, often triggered by bruising or rapid muscle growth, and the organism starts producing cytotoxins that destroy muscle. The animal can die within 12 to 24 hours. By the time you see clinical signs, it's often too late. This is why clostridial vaccination in cattle and small ruminants is considered non-negotiable preventive medicine. Three don't always get enough airtime, but they're highly clinically relevant. Listeria is fascinating from a food safety standpoint. It grows at 4 degrees Celsius, meaning your refrigerator doesn't stop it. In ruminants, poorly fermented silage with high pH is the classic source. The pH should be below 4.5 to inhibit growth. When animals ingest large amounts, the bacterium invades macrophages and actually polymerizes host actin to propel itself through the cytoplasm from cell to cell, an incredible evasion mechanism. C. pseudotuberculosis causing Caseous lymphadenitis is one of the most economically important small ruminant diseases worldwide. The waxy, lipid-rich cell wall makes these abscesses incredibly resistant to antibiotics, which is why culling is often the recommended management strategy. Pull this together. The gram-positive world is diverse, but it's organized. Morphology on the gram stain gives you the first branch of your decision tree. Then you layer in the site of infection, skin wound, joint, gut, lymph node, and the clinical context. Is this a normal flora opportunist or a true primary pathogen? Staphylococci almost always require a host defense breach. Streptococci can be both commensal and primary pathogen. Clostridia are environmental and toxin driven. Each group has a logic to it. Learn the logic and you'll know where to look and what to expect without needing a memorized list. Read the case, think about it, and then we'll work through the answer together. Think through the biology. S. equi is transmitted through direct contact and fomites. It survives for days on surfaces, water buckets, and tack, but is not a long-term environmental survivor without a host. This means strict quarantine of the index case and any horses with lymph node swelling or nasal discharge. Water sources, buckets, and tack should not be shared. Recovered horses can be guttural pouch carriers. Asymptomatic horses that harbor the organism in their guttural pouches and intermittently shed it. Endoscopy and culture of guttural pouch washes identifies carriers who look clinically normal. This is why outbreak clearance requires testing of recovered horses, not just resolution of clinical signs. The biology demands it. Read the case question, think about it, we'll then work through it together. Great question to field and practice. The key is the spore. Clostridium species persist indefinitely in soil as spores, so exposure never goes away. The animals live with the threat year-round. Vaccine-induced immunity wanes over time. Antibody titers drop, and memory alone may not mount a fast enough response when the animal is suddenly exposed to germinating spores in traumatized tissue. The disease can kill within 12 to 24 hours. The immune response has to be fast and pre-primed. Annual boosters maintain protective antibody levels against the toxins that do the actual killing. You're not just vaccinating against the bacteria, you're vaccinating against the toxins. And because most of these vaccines are toxoids, they prime for toxin neutralization, which is exactly what the animal needs. Read the question, think about it, and then we'll come back and work through it together. Think systematically. Chronic wounds and Staphylococcus together immediately raise the question of biofilm. Bacteria in biofilm are encased in a polysaccharide matrix that dramatically reduces antibiotic penetration. Minimum inhibitory concentrations within a biofilm can be 100 to 1,000 times higher than for planktonic bacteria. So the antibiotic might look active on an in vitro susceptibility test, but fail completely in the wound. Second, consider MRSP, methicillin-resistant S. pseudointermedius, which has become genuinely common in dogs with recurrent skin and wound infections. MRSP carries the MEK-A gene encoding an altered penicillin-binding protein that beta-lactams simply cannot bind. You need a culture and sensitivity that includes screening for methicillin resistance, and you need to think about whether physical debridement or surgical management is needed to disrupt the biofilm. Antibiotics alone won't clear a mature biofilm.